Okay, today what we're going to do is we're going to look at our fourth and final class of biomolecules called nucleic acids. So that would include DNA and RNA. And this is an incredibly important set of molecules that controls a lot of the functioning that goes on in cells and ultimately controls the manufacturing of any kind of compound that a cell might require. So you can think of this as we have a factory and we've got a factory going full tilt. It is building, it is producing, it is manufacturing things. So you can see in our factory here, we've got these robots building a car. And those robots have been programmed to follow intricate motions. So there's some kind of plan if you want to build this car correctly. Well, the same thing happens in cells. We have to have a plan, we have to have a blueprint, and of course, that blueprint is looks like this, right? Here's blueprint. It's got the schematic showing you all the different steps that we need to take to build something. And here's our blueprint. DNA. It also has all the information we need to build anything we need in a given cell. So it's really important that we understand the chemistry of DNA and RNA so that we can understand the processes that take place inside of a cell. So these nucleic acids, both RNA and DNA, they're polymers. Now remember, think back, polymers are large molecules. They're large molecules, but they're made up of smaller units that we refer to as monomers. So they're sort of the subunits. And we saw in the past that we have polysaccharides that were polymers of glucose, where the subunit was glucose being repeated over and over again. Well, for DNA and RNA, those monomers are called nucleotides. So we're introducing another subunit, another monomer, and for both RNA and DNA, they are called nucleotides. Now they're starting to get a little more complex. A nucleotide looks like this. It actually has three components. Now we're going to start running into things that should become familiar to you, that as soon as you look at this nucleotide, you probably could have picked out the subcomponents of the nucleotide. There's a phosphate group. Well, we've run into phosphate groups before when we talked about phospholipids. Second component is a 5 carbon sugar that is a monosaccharide, and so that should have been somewhat familiar. And then you have what distinguishes each nucleotide from one another is what's called a nitrogenous base. Okay, and it's this ring structure, and we'll look at the actual individual structures in a moment. So three components: phosphate group, sugar, and then a nitrogenous base. Well, let's look at what sugars we're talking about. Well, it depends on whether we're talking about RNA or DNA. RNA stands for ribonucleic acid. So in RNA molecules, whether it's mRNA, tRNA, rRNA, the sugar will be ribose. We've seen ribose before. We know it's a 5 carbon sugar. In DNA, though, the sugar is different. It is deoxyribose. DNA stands for deoxyribonucleic acid, and you can see on the second carbon in that the oxygen has been eliminated, and that's why it's called deoxyribose. And you can see that right here down in the corner where you have a hydrogen instead of a hydroxyl group. Okay, so if we're in RNA, the sugar is going to be ribose. If we're in a DNA molecule, the sugar is going to be deoxyribose. Now let's look at those nitrogenous bases. Right, there are actually 5 nitrogenous bases, and they fit into 2 different classes. The first class are what are called pyrimidines. Okay, and you see this 6-membered ring. You see some nitrogen within the ring. The rest of it is filled out with carbons. Well, one of the pyrimidines is thymine. Here it is. I gave you a spatial kind of model of thymine, and you can see remember now the blue dots are the nitrogens, the gray ones are the carbons, and the red ones are oxygens. And you can see the specifics then of thymine. So what you've got attached to that ring determines whether, which pyrimidine you actually have. For thymine, it's noted by this methyl group over here sticking off the side of the chain. Okay, that's one of the pyrimidines. A second pyrimidine, again, would have the same basic shape, is cytosine. And in cytosine, you can see that sticking off the side of the chain is an amine group. All right, so it's what's coming off the side. In thymine, it was a methyl group, and in cytosine, it is an amine group. There is a third pyrimidine, which occurs in RNA, and that's called uracil. And uracil again has a group that distinguishes it from the other pyrimidines, and it's down here in the right hand corner. You can see you have a carbonyl group, and you have one over on the left side too. All right, so that's what distinguishes uracil from the other pyrimidines. So pyrimidines, they are six-membered rings, a single ring. The other set of nitrogenous bases are called purines, and they have two rings. And the two nitrogenous bases that are classified as purines, first of all, we have adenine, and adenine is distinguished because it's got a methyl group an amino group. That's an amino group. Remember, the blue dots are nitrogen, so that's actually an amine group. So that's what distinguishes adenine, and is again derived from the basic purine structure. There is a second nitrogenous base that is a purine. All right, so it's going to have a different functional group coming off of it, and that's guanine. And guanine, you can see up here, we've got a carbonyl group, and we've got an amine group. So when you're distinguishing the nitrogenous bases, they have a fundamental ring structure that they are derived from, and kind of like when we're talking about steroids, whatever you attach to those rings, that's what determines what specific nitrogenous base you actually have. All right, so those are our monomers. So you've got their nucleotides, they've got a phosphate group, a sugar molecule, and then one of these nitrogenous bases. So we're going to start linking them together. And here we see four nucleotides connected to one another. Now, in this particular diagram, they're not actually drawing the structure of the nitrogenous base, but you can see along the strand here that you have the sugar. If we start at the bottom of the screen, you have the sugar and the phosphate group of the first nitrogenous base, and it's going to link to the phosphate group is going to link to the sugar ring that's part of the next nucleotide. And this is going to be, again, these are called phosphodiester bonds, and they are formed like a number of bonds we've seen through a dehydration reaction. Now, we've run into multiple dehydration reactions, but what distinguishes them is we have different molecules being linked. So we must have different enzymes that are specific for catalyzing the formation of those bonds. So don't think that the dehydration reaction forming a glycosidic bond or forming a peptide bond are going to be the same enzymes that are going to catalyze the connecting of these nucleotides. So you can just see you have alternating sugar phosphate group of the first nucleotide, then the sugar and the phosphate group of the next, then the sugar and the phosphate of the next, and so on and so forth. So if we wanted to break these up, we would have to have a different enzyme that would add water back across that phosphodiester bond. All right, now this diagram though gives you a little more detail, and you can see that sort of the backbone. You can kind of liken these strands to a ladder, and in this case you can see I've got a picture of half a ladder. So if you think of the backbone of the ladder being formed by the sugar and the phosphate group of each nucleotide, And here, since it's only a single strand, we've only got half of a rung. And the rungs are the nitrogenous bases that are sticking out from the side of the backbone. Okay, so each nucleotide had its own nitrogenous base. All right, and if we had an RNA molecule, that's all you'd have. You'd have just a single strand. All right, there you can see there's your first nucleotide. The second one here, again, you can see that phosphodiester bond that connects it to the first one, our third and our fourth nucleotide. So it's easy to start looking at these and not get confused, because things will start to look familiar. Again, we don't want these structures to seem like just a jumble of letters. You should be able to start locating landmarks and distinguishing subparts of each larger structure. All right. Now, if I'm going to make it DNA, we know DNA, I think you have a sense that DNA is actually two strands connected together. So those half rungs have to become a full rung. And to do that, we've got to have those nitrogenous bases. They've got to link up with a complementary nitrogenous base from a different nucleotide. And here are the pairings. Here are the different nitrogenous bases. They're going to connect, and they're going to connect with something again that's familiar by this point. And those are hydrogen bonds. So the rungs, the half rungs that eventually form a full rung, are formed through hydrogen bonding. And the complementariness comes from whether you've got two hydrogen bonds or three hydrogen bonds. And so thymine can only link up with adenine. That's its complementary nitrogenous base. Cytosine bonds with guanine because they can form three hydrogen bonds. So the rungs of our double strand, what's going to eventually be a double helix in DNA, is actuated by these hydrogen bonds forming. You can't have a non-complementary nitrogenous bases link up. Okay, and you can see this graphic shows that clearly. That if you don't have the right number of groups to form the proper alignment to form the hydrogen bond linkages, they would not connect with one another. So adenine with thymine, guanine with cytosine. Now, in RNA, there is no thymine, so we would have uracil instead. Adenine would be linking up with uracil. Here now you can see the detailed double strand, and you can see how the nitrogenous bases are forming the hydrogen bond linkages in down the center there. They're sort of in these pink X's that you can see are highlighted across the very center. And again, the complementariness comes from matching up the proper number of hydrogen bonds that can form. Okay, so DNA and RNA, both made up of nucleotides. RNA, their nucleotides have ribose in them rather than deoxyribose. When you RNA does not have thymine, it has uracil. And all RNA, no matter what variety we are discussing, they're single strands, whereas DNA is a double-stranded molecule. DNA, it was a big deal being able to discover and detail out its structure, and that happened back in the early 50s. You may have heard of Watson and Crick. Their key work in analyzing X-ray diffraction crystallography pictures that allowed them to figure out exactly what was the structure, what was the molecular structure of the genetic material in cells. That's all the way back in 1953. All right, so DNA is double-stranded, RNA single-stranded. Now, this double-strandedness, it also is related to things that we've talked about before. Why be double-stranded? Why does it have to be spiraled like that? Well, it has to do with those nitrogenous bases. Those bases actually are to a large degree, hydrophobic. They're trying to get away from the aqueous environment that the DNA would be encountering. And so on the outside of this spiral would be the sugars, which we know are hydrophilic, and the phosphogroups, groups, which are also hydrophilic. So they're facing outward, whereas the rungs, the nitrogen spaces, are facing inward. And the twisting helps to reduce even further the connection, uh, contact with any water that might be present. So again, things that we've talked about recently come into play, having a fundamental impact on the shape of this molecule and its characteristics. All right, so those are the compositions of RNA and DNA molecules. Now, DNA, vitally important. Incredible amount of research goes into fully understanding DNA, and a key aspect, other than passing genetic information from one generation of cell to another generation, is its role in protein synthesis. And so you see here in this diagram a cell with a nucleus. The DNA obviously is located inside of the nucleus. And let's go through the steps. Let's see what's involved here whenever we want to synthesize either a peptide or a protein. Well, the first thing we've got to do, these molecules are huge. DNA is very, very large. So if I want to get to the information contained in it, the first thing we've got to do is we've got to let those chromosomes, which is composed of the DNA, we've got to let it unwind a little bit. And you can see in the diagram, it's unwound a little bit. Now we've only unwound a portion of it, only the part that we're interested in that serves as the blueprint for the peptide or the protein that we're trying to synthesize. So we unwind it a little bit, we expose it, and we transcribe it. So in other words, we're going to copy. We're going to copy the information that carries the blueprint for, again, whatever peptide or protein we want to synthesize. That's called a gene. So any portion of a DNA molecule that codes for a particular peptide or protein, that's called a gene. You've heard of genes, that's what they're referring to. All right, well, how do we do that? Well, what we do is we form a mRNA, a messenger RNA molecule, making complementary strand that matches up with the original code in the DNA. And then that information, so we got to get that. We're not going to make the protein in the nucleus, it's going to be made out in the cytoplasm. So the mRNA is going to then leave the nucleus, and it's going to go out to a specific structure in the cytoplasm, and that's called the endoplasmic reticulum. So you can think of that as sort of the factory where we're going to take this blueprint that we just created, we're going to take this mRNA, and we're going to start to decode it. And that's what's going to happen in the endoplasmic reticulum. There are structures called ribosomes. They just go right along the length of that chain of mRNA, and it's going to start reading the code. And every 3 nitrogenous bases, which is called a codon, will code for a specific amino acid. So every amino acid has at least 2 or more codons, actually, that code for it. And so we have a second RNA called transfer RNA. You can see they sort of have a clover leaf sort of shape, and they are complementary, and that's why we call them anticodons to the mRNA, and they bring along the correct amino acid, and then we just start linking them together, and you can see the little chain of amino acids starting to grow coming off of the ribosome. So here is a chart that actually gives you the codes for the different amino acids, and you can see every amino acid has at least 2 codes. Some of them have up to 4 codes that code for them. You also notice a couple of other things here. There are also signals within the gene that tell us when to start reading the code, start reading the information. So it's sort of a signal, well, here's the beginning of the blueprint for whatever peptide or protein I need to manufacture. There are also codes to stop reading and end, and we would be at the finish of the production of the peptide or protein. Here's just a little bit of detail about the endoplasmic reticulum. You can see it's adjacent to the nucleus, so when the messenger RNA leaves the nucleus, goes right into the endoplasmic reticulum, and the ribosomes start reading it. And here you can see a depiction of that process. This is a really good diagram because it shows you that the ribosome is moving along the length of the messenger RNA. It's reading the code, the codons, every three nitrogenous bases. You can see the anticodon, the transfer RNA coming in with the correct amino acid that was coded for, and then you can see stringing off the top left of the ribosome, you can see the growing peptide chain forming as these peptide bonds are formed between the amino acids. mRNA and tRNA, transfer RNA, they are single strands and they have their own special characteristics that I referenced. So this slide really just kind of summarizes that for you. One other thing we need to realize about DNA is every time a cell divides, as we produce more cells, we need to take the information, the genetic information in the nucleus, and we need to make sure it get passed on to the daughter cells, the cells that result from the division. So DNA has to be able to replicate itself. And you see in this diagram that the double helix of the original DNA has opened up, and you can see a complementary strand is being formed. So at the end of this replication process, we will have two exact DNA molecules, and one will get sent to each of the daughter cells. So we maintain the passing of genetic information from the original cell to subsequent cells. To kind of wrap this up, these molecules, they're enormous. A DNA molecule has 3 billion nucleotides, enormous molecules. That is about, if you were to actually have it stretched out in a linear fashion, that would be over 5 feet long. And yet we got to get this thing compressed down into that tiny nucleus of the cell. And so that involves a lot of twisting and tightening of the double helix, and you can see as you move on the left hand diagram, as you move from the bottom to the top, it's getting wound tighter and tighter and tighter, so it's really highly coiled by the end, and that's what makes a chromosome. And you can see on the right hand side a photomicrograph where the chromosomes have been stained so that they stand out, and you can kind of get a feel for that tightly coiled structure. This these are electron micrographs, and you can see high details of the chromosomes, and you really get a sense from these images of the highly coiled nature of chromosomes. These are enormous molecules, lots of information stored away in the DNA.